Hello everyone. Today I'm going to present about vaccine development as a part of our medical biotechnology module. In these presentations, I will cover vaccine history, technology platform, COVID-19 vaccine, edible vaccine, the development process, and emerging technologies. A vaccine is a biological preparations designed to provide immunity. It may contain vegan or killed pathogens, toxin, or even modern components like mRNA. The immune system recognizes the vaccine and remembers it for future defense. Vaccines have helped eradicate diseases like smallpox and control influenza and polio. Now let's quickly look at some of the most important moments in vaccine history. Vaccines are made using different technologies. Here are some of the most common types. First one, live attenuated vaccines. These use weakened versions of the germs. They give a very strong immune response, but they are not safe for people with weak immune system. An example is the MMR vaccines, inactivated vaccines. These use a killed versions of the germs. They are very safe, but we often need booster shots to keep the immunity strong. The IPV inactivated polio vaccines is an example. Subunit vaccines. These use only small part of the germs like protein. They are very safe and specific, but the immune response can be a bit weak. The HBV vaccine is an example. Next one, toxoid vaccines. These use a deactivated toxin from the germs to create immunity against the toxin itself. Tetanus and diphtheria vaccines are examples. Viral vector vaccines. These use a harmless virus to deliver instructions to our cells. They give a strong response but can be costly. The Ebola vaccine is one example. mRNA vaccines. There are newer types of vaccines that uses genetic materials mRNA to teach our cells how to make a protein from the virus. They are very fast to produce, but they need to be stored at a very cold temperature. The Pfizer and Moderna COVID-19 vaccines are the most famous examples. This slide shows how different technologies were used to create the COVID-19 vaccines we all know. The Pfizer and Moderna vaccines are mRNA vaccines. The Johnson & Johnson's vaccine is a viral vector vaccine, and the Novavax vaccine is a protein subunit vaccines. Vaccines come in different forms and they are given in different ways. Vaccines are present single dose virus, pre-filled syringe, or multi-dose virus. They can also be administered orally or nasally. For example, rotavirus is given orally, influenza intranasally, MMRV subcutaneously, and COVID-19 vaccines intramuscular. Edible vaccines. Did you know we might one day eat our vaccine? This is a new and promising area called edible vaccines. Edible vaccines use genetically modified plants to produce antigens that can trigger an immune response. Examples include potatoes, bananas, rice, and tomatoes. Advantages. They are safe, cheap, and you don't need a needle to get them. They also don't require cold storage, which makes them easier to transport, and they could potentially protect against multiple diseases at once. Disadvantages: the immune response can be a bit weak. It's hard to control the exact dosage, and there are many regulations to follow. Making a new vaccine is a long process that has many stages. Walk through each steps. First one, exploratory phase. This the research stage. Scientists spend 2 to 4 years identify a good antigen to target. Second one, preclinical phase. Third one, clinical trials. It has a three phases: phase 1, phase 2, phase 3. Fourth one, regulatory review and approval. After the successful trials, the vaccine is reviewed by a regulatory body to get approval for public use. Next one, production scaling. One approved, companies start making the vaccine in large quantities. Next one, quality control and post-marketing. Even after approval, the vaccine is continuously monitored to monitored for safety and quality. Beyond the main types we discussed, there are also some new technologies in development. Virus-like particles, VLPs. There are particles that look like a virus but don't contain any genetic material, so they are very safe. Examples include the vaccine for HPV and HBV and nanoparticle vaccines, NPS. These use tiny particles to deliver antigens. They are cheaper to make and more stable, but they can sometimes lead to a weaker immune response. When the designs vaccines, we must consider safety, efficiency, cost, storage, ease of administrations, number of doses, and the durations of immunity. In summary, vaccines are one of the greatest achievements in medicine, from genus cowpox vaccines to modern mRNA vaccines. They continue to save millions of lives. New approach like edible vaccines and nanoparticle systems show that the future of immunizations is existing in innovations. Thank you. I hope this